so much.
Who's introducing the next lecture?
Me, I'm here. Indeed, I was checking.
You're tricking me, eh?
Yes, so the next speaker is actually already connected, so I'm just gonna show briefly a couple of slides, and then I'm gonna leave the floor to Michel Thiebaut de Schotten. So Michel is really, I mean, I cannot think about a most successful researcher than him. He is leading the Brain Connectivity and Behavioral Lab that was founded in Paris. And also now he's in Bordeaux, leading at the head of the Neurofunctional Imaging Group at the Institute of the Maladies Neurodégénératives of the CNRS in France. Michel won the ERC Consolidator grant with about 2 million fundings, and I worked this project when I was in Bordeaux, looking to the disconnectome and the impact of disconnections to treat patients with a stroke and actually and also predict the clinical symptoms there. So looking again to a kind of initial stage of the evolution of the disease. And actually now Michel published this paper that actually, I guess also mitochondria are really important in the metabolism, and was published in Nature, and as Nature has been Nature. So it's and it's been defined as a groundbreaking achievement. So the first map of human brain mitochondria, and I'm really glad that he's here, connected online from Bordeaux, to talk us about this great project and this new vision of the brain metabolism. And now I'm gonna start his streaming.
Mitochondria is able to convert oxygen and glucose into energy through the Krebs cycle inside the cell. And that provides us with the energy we need to move and do everything we do every day through an aerobic process of energy. Can I have the next slide? And so energy matters, as you can imagine, but it matters particularly for the brain, and particularly for the human brain, because in humans, the brain, despite representing about 2% of the mass of our body, it's going to use 21% of the energy, and this is just at rest. And the variation from rest to activity is just about 5%. That means that in spite of being small, our brain requires a lot of energy, and the way those mitochondria are going to behave or can be sick or stressed might affect our well-being, our mental health, and the rate of our aging. Go for the next slide. Quick information, if you didn't know, mitochondria have their own genetical code. It comes from your mother, and the mother of your mother, and the mother and of the mother of the mother, up to Lucy in Africa. And so they are transmitted matrimonially. And the DNA is actually different from the human DNA. It's a little circle like a donut. It's easy to spot on post-mortem exploration of the tissue with very high resolution. And I'll get the next slide. So, because I got interested into mitochondria, and I work with this team in Columbia University in New York to that subject matter. So we're doing it in the tissue. They wanted to work on the brain. We started wondering whether mitochondria can be changed by the interaction with the environment, or could change our interaction with the environment when themselves are sick or depleted or increased. But the problem is, up to today, we didn't have a good way to map and understand, explore mitochondria in the living human brain. Take the next slide, please. Well, this is a little bit the illustration of what I just told you. If you stress people, whether this is a physical stress for the muscles, the mitochondria that will be in the cell of the muscles, or an experimental stress that will stress your brain, this is actually bad for the mitochondria. They don't like it, and so they reduce their density and their efficiency. And this, in turn, on the long term, the hypothesis is that it will have an impact onto your well-being. It will have an impact onto the aging of your brain. And so if you believe in that theory, then maybe mitochondria might be one of the earliest biomarkers that you can extract from a biological system, from the brain, in order to predict how people are going to become eventually with a neurodegenerative disease. And if you can manipulate this biomarker via interaction with the environment, maybe you can prevent this aging trajectory. On the other hand, well, that's still good, okay. But on the other hand, if the mitochondria are sick because of a biological disease, you can observe in mitochondrial disease strong impairment into the functioning of the brain and the behaviors. It's a study that we have at the moment carrying on in New York. Next slide, please. As I mentioned before, we do not have any way to look at the mitochondria in the human living brain, and this is because there is no imaging strategy to see it so far. So we thought eventually, because we have a large spectrum of neuroimaging that we can acquire in the human brain, and we can look in postmortem tissues, the mitochondria, can we try to bridge a gap between the two? And to do so, we had to create this unique data set, which is a slice of brain that we cut into low voxel of 3 by 3 by 3 millimeters. Next slide, please. So you can see on the right, it's Eugene. He's a researcher that put in place all this post-mortem cutting. He's wearing like a coat and a hat because of, obviously, he had to stay in that room that is freezing cold for so much time in order to make it work. On the left, you have the sample where we already removed some of those real voxel of postmortem tissue one by one with our chisel, being very, very careful of labeling each of those voxels with a specific coordinate in order to be able, in the end, to reconstitute our sample in a digital space. Next slide, please. So, to summarize a little bit what we did for this part of the experiment, we took one slice of the brain that would be on the top left, then we cut it with this fantastic machine in little cubes of 3 by 3 by 3 millimeters. And those 3 by 3 by 3 millimeters, you can put them into a tube and do mitochondrial enzyme assay, respiratory, and RNA sequencing on it, which then you can put into a matrix with numbers corresponding to this biological measurement. And because those biological measurements correspond to coordinate in space that we kept since the beginning, you can project it back into the brain, stretch it into the MNI space, and then try to predict those values with neuroimaging and project it onto the entire brain. Now, there was no difference of weight really between those voxels, the amount of protein was the same, and the number of DNA copy was also the same, so they're quite comparable, more or less. Next slide. So for each of those little voxels, you can extract different kind of values. You can extract different level of complexes of the respiratory chain, which are the complex 1, complex 2, and complex 4 that can be different from one mitochondria to another. Those are essential in order to convert oxygen and glucose into energy, and if you do this magical equation that you can see on the right, you can calculate the tissue respiratory capacity that corresponds to the surface of those different complexes, how much surface they do cover to process the oxygen and the glucose. And then if you look at the mitochondrial DNA, you can quantify the density of mitochondria inside your tissue by counting how much DNA of mitochondria you have in your tissue. And if you divide the tissue respiratory capacity that I gave to you by the density of mitochondria, you have an estimate of how well each of your mitochondria can breathe and convert oxygen and glucose into ATP in the brain. Then you use those values, you project them back onto the 3 by 3 by 3 millimeter sample space, and you can start seeing emerging a pattern that is pretty clear that you have way more mitochondria in the gray matter than you have in the white matter by 2 or 3-fold. That means that the conversion of oxygen and glucose in the gray matter is way more important than in the white matter. So if you're looking at fMRI in the white matter, you gotta know you get 3 times less signal just because of this biological fact. And we played around onto this comparison that you can see at the bottom, but for the purpose of time, I'll move to the next slide. Thank you. And then we started looking at pattern of the different regions of this postmortem sample. If you look at region in the brain, according to whether you share those regions with other species, whether you share it with primates, with mammals, with other earlier like crocodiles and fishes that we separate from longer time ago through evolution, you can reorder those regions according to their phylogenetical age. And there will be the part on the right, and if you look at the order of region according to their phylogenetical age derived from comparative anatomy, you can see that there is a direct correlation between the phylogenetical age, evolution of the brain, and the mitochondrial respiratory capacity and the mitochondrial tissue respiratory capacity. It's very strong correlation there. It means that with evolution and the new regions that appeared in the brain, we started developing regions that are more greedy in terms of energy needs and optimize the mitochondria in order to convert more oxygen and glucose into energy. Now the mitochondria are not the same everywhere in the brain. There is a part at the bottom where we did some more extensive characterization of the mitochondria according to the type of cell that are inside the 4 voxels that we more intensively explored. And you can see that oligodendrocytes have a different pattern of mitochondria from microglia or inhibitory or excitatory neurons. This is a representation that is called a UMAP embedding, where cells that have a similar profile will be clustered together, and cells that have different mitochondrial profile will be spread apart. Next slide, please. And that's where the neuroimaging action is coming into place. So you can see at the top, this is the postmortem slice. The idea is really to put anatomical landmark onto that slice and report those anatomical landmark into the stereotaxic space that you have in the bottom left of that slide, which is in D, and create a field of deformation that then you can apply to a grid when each of those little voxels have a specific number in order to be able to collect in the neuroimaging space values that are matching the postmortem space. So you have corresponding matrices between postmortem histology, biology, cellular imaging, and neuroimaging. And if you have a corresponding matrix, you can create a model. You can do very advanced model. I'm more a classical guy, so I went for hierarchical regression with backward elimination. That was better or as good as the most advanced model, by the way. And this approach gives you how much of each of the neuroimaging features you have are good when you put together to predict the post-mortem measurement of C1, C2, C4, mitochondrial density, tissue respiratory capacity, and mitochondrial respiratory capacity. It's a recipe of linear combination of neuroimaging. You have the recipe on the right part, this is a table, as well as all the different features that we've been exploring. Those are not the same brain. We use templates from the MRI shared database to predict one single postmortem brain. And even if they're not the same brain, you'll see in the next slide that the correlation were pretty good. If you predict at the voxel level, and here we predict voxels that were kept aside, 20%, so there is no overfeeding of the data, 20% that were never used to learn the rule how to combine our neuroimaging to predict those mitochondrial features. At the voxel level, you have correlations that are going from 25% to 35% for those features. And this is because they're not the same brain. But if you combine those information into brain regions using simple atlases, then the differences, individual differences, flattened, and you have this strong correlation that emerged that is going from 70% to 87% of shared variance between the predicted values from neuroimaging and the observed value from mitochondrial feature extraction outside of the sample that has been used to learn. If it works for voxels we didn't see, it should work for the entire brain. And that would be the next slide. So once you have the recipe, you can apply this recipe to the whole brain and map on the entire surface of the cortex, on the lateral view, medial view, and onto the white matter, well, onto the white matter connections, because those connections are going inside the cortex as well, the values of mitochondria. As you can see here, we're kind of going in the same direction. If you have a high mitochondrial respiratory capacity, you have a lot of C1, C2, and C4, and your mitochondrial density will also tend to be higher. Tissue respiratory capacity will also be higher. Next slide, please. Then we were wondering, is there like a really structure behind the organization of those mitochondria? So we created a UMAP, I mentioned it before. We take all the voxels of the brain, we put them into a two-dimensional space where voxels that share the same mitochondria are close together. The ones that share different mitochondria are spread apart. What you can see, like in the blue and red UMAP, is that automatically you can spot that the gray matter probability is a main factor explaining the segregation between the type of mitochondria. But also, if you start looking, and those are like little square with purple dots, if you start looking at structure in the brain, given structure in the brain tends to also have a specific signature for mitochondria, and that works because those vocal cells are close together, but that also works for long white matter tracts that can go from the front to the back of the brain, and they'll be located in a very special localization into this embedded space, meaning that if you have an inferior fronto-occipital fasciculus that runs from the frontal lobe to the occipital lobe, all along the inferior fronto-occipital fasciculus, you'll have a similar signature of mitochondria that is specific to that white matter tract. At the bottom right, we had the chance to have an extra slice of brain in the occipital lobe where we could verify whether our prediction of mitochondrial feature could extend to another sample of the same brain, but in a different part of the brain. You can see that we still had a correlation between the predicted and observed mitochondrial features. Next slide, please. Just to give you an idea, so we go from 730 voxels that were quantified for different mitochondrial features, and through the learning approach and the neuroimaging, we can increase also our resolution. You can see on the right, you see sharper details, of course, of this mitochondrial distribution that shares the same property as the original data set, such as, and that'll be in the next slide, the relationship with evolution that you can measure in neuroimaging in two different ways, either by looking at the monkey to human expansion, which is called phylogenetical map, but we can all agree that monkeys had their own evolution going on. They're not like ancient humans. Yet, this is the best way we can do. And gray matter variability, because regions that are more variable, more different from one individual to another, tend to be more recent on the evolutionary tree. And you can see here that the correlation between the predicted mitochondrial maps and those viability map and monkey to human expansion, this correlation stays. So the predicted map shares the same relationship with evolution than the original raw measurement of mitochondria. Next slide, please. And those bring us to the conclusion here, where we can ask where we're going to go from there. There's like several things that we will expand from there. We can expand the same predictive model to other brain samples, although you may have lost track by now, but those 750 voxels that were explored were already costing about $150,000, so it's really expensive. It takes a lot of time to do, but you know, we'll put it in the grant, I guess. Investigate patients with mitochondrial dysfunction. This is what we're currently doing at Columbia University, and we did find specific differences. Apply the same model to other histological and cellular biology metrics. We have a paper that we're polishing up where we expanded this to cytoarchitecture. So we can predict the cytoarchitecture and do, for the first time, digital cytoarchitecture from neuroimaging. And in the second part, we were thinking about making a relationship between estimated mitochondria and variation in cognition in healthy controls and in pathological brain. And finally, one last line of exploration that we're currently doing is trying to manipulate these mitochondrial expressions through photobiomodulation, because they do have an appetence for specific light and tends to increase their activity and multiply when you shine some bright light on them. Thank you very much.
Thank you. Thank you, Michel. You can hear me. You can see me here, okay?
Yeah. Yeah.
Okay. Did anyone have any question? Maybe. Yes, they are coming up.
Okay. Hello, can you hear me? Or can you see me also? Yeah.
I can see you, but I can see also. I can see.
Oh, you can see me to the back.
Yeah, but I can see the front as well. I get both views.
Anyway, it's just you can hear me, it's enough. I'm really impressed. Thank you very much for this presentation. One of the questions is really pretty private. I mean, we are studying a mitochondrial disease which is called MELAS, mitochondrial encephalopathy with lactic acidosis syndrome, especially for the heart, because the heart as well hosting a lot of mitochondria. Do you think that such a technique could be useful to understand how the heteroplasmic on mitochondria may be involved in the ATP consumption, ATP reduction, and therefore also the quality of the ATP production, especially also in the brain, but in the other organ as well?
Yeah, that's an excellent question, and we're publishing new results that I didn't have the time to present exactly on that subject matter. So we did explore mitochondrial disease in the brain using this approach and bringing those maps to the individual level. And one thing that you can spot right away, not in MELAS, because it tends to not have a good compensatory mechanism, is that the complex 2 in the single dilation, the complex 2 actually increase a lot in mitochondria. And this is because the complex 2 is encoded by the gene of the nucleus of the cell. It's not encoded by the genes of the mitochondria itself. So you can have compensatory mechanism from the interaction between cell and mitochondria that you cannot have for other complexes that are encoded by the genes of the mitochondria directly. At least that's how we interpreted the results.
Okay. Thank you.
Thank you, Michel, for your nice talk. Now, it's interesting what you said in your last slide, that they try to apply to pathology. And one thing which comes to mind is that for Alzheimer's disease, which is the condition I study most, there is also a mitochondrial hypothesis. And one set of our studies looking at reprogramming from fibroblasts, astrocytes from people with sporadic AD. An interesting finding was that the mitochondria of the AD patients did have impaired respiratory capacity. And interestingly, that parameter correlated with poor cognition, especially in fluency. So it is a nice parallel between what we see in pathology and what you actually see, that respiratory capacity is the parameter that actually correlates with cognition. So I found that very interesting.
Thank you. Yeah, this is absolutely true. I didn't work on mitochondria before, and I started like 2 years ago, so my knowledge remains limited. But in the reading I've been doing, I was surprised to see that actually mitochondria are the ones that are deciding for the apoptosis of the cell. They just give up. And then there is plenty of studies that also show that you have more plasticity if you, that's in rats, but if you inject healthy mitochondria into the damaged cells. There is also the studies and more discussed that shows that aberrant cells can also be limited by neighboring mitochondria that can protect from the invasion from those aberrant cells. So there is a lot of promising things we can do, and I'm really excited to see that now we can eventually study that with neuroimaging. It is also possible, and we discussed that in Bordeaux, because there is a big center studying the heart to decline the same model to measurement of the heart. So it's a matter of time and students to do it, but the principle it's feasible. And then finally, we also have some additional recent results that I didn't present because I finished it yesterday, but we have preliminary results showing that those predicted mitochondria, particularly the complex 1, that we can also measure with a specific PET ligand. So we do have a correlation between the two. So it's very encouraging. It seems like we can really see it. And now the next step will be to try to use it to understand better how it can work as a biomarker or early biomarker or something that we can manipulate to prevent neurodegenerative disease.
Not only as a biomarker, but potentially as an approach to treat that deficit, because there are medications that you can actually use to improve respiratory capacity. Yeah, very interesting.
That's right.
Thank you.
You also have like TPVM, which is it's a light photobiomodulation that also increase respiratory capacity of the neurons. So it's a very interesting field. Excited.
Yeah, it's an exciting time. Thank you.
To do that.
I have a little question. Actually in my first postdoc, I worked a bit with mitochondrial disease in the brain, and we were seeing, for example, in the Leber pathology, all the white matter hyperintensities in the like close the optic radiations, where the metabolism, I love to see that the long-range connections, as the IFOF were on this one side of the UMAP, others on the other side, and yeah, I mean, you're really specific and biological like focus, but I was just curious to know if you think somehow also to see at white matter hyperintensities, or it's not really in the picture, because it seems that when you have a dysfunction, not just vascular, but also from the mitochondrial standpoint, and you see that in people with rare mutations of the mitochondrial, you have as a phenotype in the brain these upper intensities, and yeah.
That's a very good point. It needs to be explored, because obviously we cannot pick a part of the brain of those patients.
Yeah,
they're
that's
still
true.
alive. So
yeah, maybe like we need banks, brain banks.
Then, so this approach, so we now replicated it like in 3 different centers, and the approach is replicable. It's just a matter of having a group of 10 healthy controls match with a postmortem sample in order to build the recipes that work. So combination of neuroimaging that works with your scanner to produce those maps. That's all there is to do. And then you apply the same equation to individual neuroimaging.
Yeah.
But it does require a good 45 minutes, 1 hour of acquisition, because you need diffusion structural and resting state MRI.
Okay.
We did not have the opportunity yet to explore other modalities, nor to do all the physics work to see whether we could do some specific measurement with the MRI scan that would reduce that time. But that's all future developments.
Oh yeah, but I think it's great, really groundbreaking, as the Nature editor said. Are there any other questions? Otherwise, we can leave Michel to his other. Okay, I think, yeah, there was a long day here, are all a bit tired, I guess. But thank you so much. And
thank you so much for the invitation.
Yeah, I hope to see you in person next time in Parma. Bye-bye.
Bye.
Okay. Yes.
So the program says closing remarks. We're a few people in the room, so I think it's going to be very, very, very short. I'm happy about how it went. I think the experience must be repeated, and I also love the multidisciplinary aspects. So I also appreciated the idea of keeping the humanities in the morning and then the hard science in the afternoon. However, maybe next time we can actually mix it up even a little more, and you know, having both humanities and STEM morning and afternoon, if we repeat something like that, so that everybody learns something. Maybe if we do that, we can kind of like tone down a little the technicalities and make it a little more divulgative. So it was perfect today because the audience was matching the presenters, but if we mix it, maybe keeping it understandable for everybody would be useful, which in the end makes of this event also a public engaging activity, which is also accounted for in the funding of the university. So thank you very much for everything. I learned a lot, and also really thank you, because once again the whole initiative started from you, and I think this is really something to be appreciated.
On behalf of Lia and Adele, I want to thank first of all of you that stayed until the end. Professor Del Rio for chairing the afternoon, and both Professor Miragoli and the Professor of Lia, that I forgot the surname. For being here and supporting us, and the whole University of Parma. So thank you.
Yeah. So you did right. So if you have a chance to get tenure, this was actually well played.